think we're getting started. Yeah. Oh, all right, you got me a bigger one. Okay. We're getting started. Welcome to your grand round. So good afternoon, everybody, and welcome to Grand Rounds. It is my pleasure today to introduce Dr. Laura Crotty-Alexander as our speaker. And it's a testament to her popularity that we've managed to get more than 4 people into the audience today. So hooray for you, Laura. So Dr. Crotty-Alexander is an associate professor of medicine in the Division of Pulmonary and Critical Care Medicine and section chief of Pulmonary and Critical Care at the VA San Diego. She completed her undergraduate and medical degrees at Duke University, followed by internal medicine residency and pulmonary critical care fellowship training at Harvard, and postdoctoral training here at UCSD in the lab of Victor Nazay, if I got that right. She's conducted research at Duke, the NIH, MGH, and UCSD, where her research focus is on the health effects of e-cigarettes and vaping, and the topic of her talk today. She's had considerable funding from the Tobacco-Related Disease Research Program of the state of California, as well as NIH, is co-PI of the recently funded Pulmonary T32, yay, as well as PI on a K24. She has published widely in the areas of e-cigarettes and tobacco exposure, asthma and inflammation, neutrophils and host defense, as well as medical education. In addition to being a triple threat, she is a mentor par excellence, having received the Excellence in Faculty Mentoring and Research from the Department of Medicine in 2023, and is also very active in the JEDI space. So I would actually call her a quadruple threat. Today, she'll be giving grand rounds entitled E-Cigarettes from Lung Injury to Tumorigenesis, Mental Health Disorders, Altered Immune Responses, and Beyond. Please join me in welcoming Dr. Crotty-Alexander.
Thank you, Zia, for that very kind introduction. I know we were told to say something personal about ourselves before we started, and I really struggled about like a story. I feel like there's so many. I guess one thing that I always think of when I try and figure out why I picked pulmonary critical care was as a medical student being in the NICU in the middle of the night and having a gentleman actively bleeding out, and there's like literally just blood everywhere. And I'd never been in that situation before. We had a Minnesota tube in place, so I had the football helmet on, and it was like so dramatic. And I remember talking to the family and watching these master, clinicians, handle the situation, and just being inspired by, the end of life, doing everything you possibly can and helping the family come to terms that, they're not going to make it. So for me, that was like a very personal experience, and it really sort of, I feel like, it's highlighted anytime I think about my path into critical care. Very different from e-cigarettes. So e-cigarettes, usually there's no bloodbath except when we're doing our mouse experiments, which I'll talk about a little bit. I have not taken any money from the tobacco industry. They haven't offered me any, so I'm a little bit upset about that. And I thought about like, what do I really want our clinicians in particular walking away from this talk with? So I picked out like 5 things, and I'm hoping to convince you of each of these things by the end of the talk. So 1, that e-cigarettes cause lung disease. 2, that they're going to cause cancer. 3, that they affect cells and organs across the body. 4, that dual use of combustibles with e-cigarettes is the worst thing ever, and it's going to have the worst health effects. And 5, that if your patient is inhaling one thing, there's a very high odds that they're inhaling something else, and maybe even 3 things. And whenever I start talking about e-cigarettes, I put it in context of combustible tobacco, which is still, the worst thing ever. Kills 8 million people per year now, and about 500,000 of those in the United States. So combustible tobacco, I think we can all agree, is very, very terrible. And e-cigarettes, which are a tobacco product, came into play, the modern form, in 2003, when they were invented. They didn't enter the international market until 2007, and then they didn't get really, really popular until around 2015. They've evolved a lot, and so it's been very hard to study them because every couple of years a new generation comes along, and so different materials and different ingredients. So I show you in the lower left-hand corner what they looked like when they first got to the United States. They looked a lot like cigarettes. And then in the middle of the bar, we see these beautiful purple and yellow and red vape pens, and then the big gray box mods. But now in 2023, the vast majority of devices are the pod-based devices. So that row of ones at the top that look like USB drives. Each one of those contains the nicotine that is equivalent to a pack of cigarettes. And then the bigger ones below that that are very colorful, those hold the equivalent of 13 packs of cigarettes worth of nicotine. So that somebody who's carrying around one of these, like, is basically carrying around 13 packs of cigarettes. So when you ask somebody, how much they vape, it really depends on the device they're using and how quickly they go through it. So when we do very specific questionnaires, we drill down on that so that we can get a real good idea about how much nicotine they're consuming. But outside of nicotine, there are some other things that are in every single e-cigarette. So propylene glycol, anybody who does bench science has probably used this to dissolve chemicals. Glycerin, also called vegetable glycerin or glycerol, is very similar to propylene glycol, but it's thicker. It makes the liquid thicker, and it makes the aerosol more visible when you exhale. Turns out that base nicotine really burns on the way in. And so the early generations of e-cigarettes use base nicotine, but the Juul makers hired chemists who figured out that if you switch base nicotine to nicotinic salts, it goes down a lot smoother. And so you're able to crank up the level of nicotine to sky high levels, 59 milligrams per mil. And so that's why our modern e-cigarettes deliver very high doses of nicotine because of that chemical change that they figured out. And then flavorants. So all e-cigarettes basically have flavors because we tried to have our human subjects vape a non-flavored device, and they all said it was hideous. That tasting propylene glycol, glycerin, and nicotine is horrible. So there's thousands of flavor chemicals that are used to flavor these devices. I think it's really interesting that, they entered the country in 2007, and the FDA actually did try to block them in 2009. But unfortunately, e-cigarette companies sued the FDA, said that they didn't have authority over these devices because they weren't being used for smoking cessation. And so they lost that battle, and so e-cigarettes flooded into the country, and as I said before, they are a tobacco product. So 2015, they're getting really popular. 2018, 85% of young adults 18 to 29 years of age had tried an e-cigarette, which is just sort of mind-blowing to me. But if you look over age on the horizontal axis, if you go to people who are 65 plus, at that same moment in time, only 22% of them had tried an e-cigarette. So this is something that, again, is used primarily by young adults. And we had done so well with children and young adults and getting their combustible tobacco rates down. But unfortunately, that's been completely replaced by e-cigarettes. So you see here the most recent data that 16% of high school students have used a tobacco product, and 90% of that in teal is e-cigarettes. So very low rates of combustibles. So one silver lining. And then we were also excited on the policy front, whereas in 2019, the federal government did raise the age to 21 for all tobacco products. And this led to a doubling of the number of states who enacted those laws as well. So now over 40 states have 21 as the age for tobacco products. And then the FDA got back in the battle in 2020. So it just took them 11 years to re-engage. And they decided that they were really, really going to regulate e-cigarettes, and they were going to ban flavored vape pods. So this was great that they were going to do this, but then immediately the e-cigarette industry shifted away from making the pod devices to making all disposable. So, they just flexed, and now the majority of e-cigarettes purchased and used are disposables because they are through that loophole. So here we are. Epidemic still continues. More than 3 million middle and high school students are using these tobacco products. It's really cute. My two sons are both in middle school, and they hear me talking about e-cigarettes all the time. And my older one came up and gave me a hug and said, "Mom, I'm never going to use, an e-cigarette." And I was like, "Write that down and sign it." Maybe. So on the mouse side, I wanted to share some data that we find really interesting and are trying to figure out. This work is being led by Jorge Massa Silva, who's one of our postdoctoral fellows, recently just got a TRDRP award, and Wanjun Gu, who's in the Simonson lab, but is now at UCSF in a grad program. This is our e-cigarette exposure chamber here, and you see our really cute CD1 mice. They each have like a pie-shaped wedge that they can run around in. And then we use commercially available e-cigarettes and activate them and put that fresh aerosol in there for them to inhale for about an hour or two every day. They don't like it the first 2 days, and then they love it. So getting them to run in after the first 2 days is very easy. So mouse addiction to nicotine. So these mice, we took the lungs from, isolated the RNA and did transcriptomics. And why I highlight this data here is that each line is a different immune or inflammatory pathway. And we found extremely different results based on flavorants. So these mice had the same propylene glycol, glycerin, and nicotine inhalation, but the chemicals added for flavor, such as ethyl butyrate and methyl butyrate and vanillin, those are different. And just the changes in the flavorants spectacularly altered the immune and inflammatory pathways of the lung. And so the tobacco and menthol mice are on the left-hand side, those first two columns. And what you see is a broad downregulation, suppression across these immune and inflammatory pathways. So it suggests that inhalation of these e-cigarettes in particular might lead to increased susceptibility to infectious challenges. Whereas inhalation of the mango and mint had the opposite, where you see increases in these inflammatory pathways and immune pathways, so that people who inhale those flavors might actually have increased susceptibility to inflammatory lung diseases. So just to add complexity to this world of what to look for in the community, add on that there are sex differences. So in this heat map, it's similar inflammatory and immune pathways. On the left, the female mice have broad upregulation across these pathways, and males have the opposite, so suppression. So interpreting this data would suggest that females would be at risk for inflammatory lung diseases, whereas the males, again, might be at risk for infectious diseases. And this work is Alexia joined the team in the middle there, Alexia Perryman, one of our PhDs. Ali Castaldi, who is junior faculty and trained with VIA, she took a look at our transcriptomic data to specifically try and see what's happening at the level of the epithelial cells. And so in the left upper corner, you see markers of the type 2 epithelial cells, and then in the middle is the type 1 epithelial cells, and on the right are tight junction proteins. And the first few columns are our air controls, and then the next 6 to 8 columns that look dark blue are the tobacco and menthol flavors. And the takeaway here is that her interpretation of this is that there is an impact on both the type 1 and the type 2 cells, might suggest that there's a decreased numbers of those cells, so cell death, raising that specter of emphysema. And then on the far right, with a decrease in the tight junctions, raising the idea that there's epithelial barrier leakiness, which we actually proved in a dish and published on for airway human primary epithelial cells. So this might be another piece of that puzzle. So I mentioned how I want you to walk away knowing that every part of the body is affected. And so when we do these mouse harvests, we take the brain, we take the heart, we take the colon, we take the kidneys. Sorry, Mark, I didn't put the kidneys up on the slide. But in some of the organs, we see upregulation of inflammatory pathways, such as in the brain, whereas in the heart, we see suppression of those same pathways. And then in the colon, it depends on how long you've exposed the mouse, for 1 month versus 3 months. So each organ is responding differently to these insults. And so unfortunately, it means that you really do have to look at each organ in isolation to understand what the impact will be over time. So when I go out and talk to kids and parents and everything, I spend a lot of time talking about initiation and trying to prevent people from starting because nicotine is so incredibly addictive. But another reason I really don't want people to start at those ages is that two really big, well-done meta-analyses showed a gateway effect. That if adolescents, so teenagers to young adults, start using e-cigarettes with nicotine in them, that they have fourfold higher rates of then also adding in combustible tobacco. So that's like a major red flag to us because, we really want to get rid of combustible tobacco altogether. And so this has been proven to be a real gateway effect. And this is just saying that. Another takeaway that I wanted you to have is the rates of dual and triple use. So the fact of, if somebody's using nicotine, odds are they're using something else as well. So this study, Claire Malhotra, who was a high school student at the time, she led this study and she's now in college. We found that in teenagers, the rates of marijuana use in Black, the non-smokers, so no use of any kind of nicotine product, the marijuana use rates were really low. And then in teenagers who smoked combustible tobacco in the darker gray, conventional tobacco, they were a bit higher, about 18% also used marijuana. And then the e-cigarette nicotine vapors, dual used with marijuana at about 45%. And then the rates of triple use were about 40% in the teenagers. So meaning that they smoked combustible tobacco, they vaped nicotine, and they used marijuana. So this was actually pretty shocking to me, but then going out and talking to teenagers in the community, they really report that usually between 50 to 80% of the people they know are vaping. So the rates, I think, are really underreported at the national level. Another thing that I wanted to highlight is that this is actually talking about combustible tobacco use, and that many populations are disproportionately affected by combustible tobacco, and they're identical in the e-cigarette patterns. So LGBTQ+, Hispanic populations, low socioeconomic class, non-Hispanic Black, they all have higher rates of combustible tobacco and e-cigarette use. So we were super excited when Juul started losing lawsuits over the last couple of years. And in May of this year, it was a huge win for California that Juul had extremely unethical practices of targeting those populations we talked about, and specifically targeting children and women with their colorful ads and how they reached these populations. And so, they had to pay out $462 million to New York, California, and other states. So thank you to all the lawyers who fought that good fight to try and stop tobacco companies from acting unethically. But what about cessation? So we all have our patients who come in and say that they've never been able to stop smoking combustible tobaccos. Should they switch to e-cigarettes? And we all want them to stop smoking combustible tobacco. So I really struggled with this. And this is actually the point at which I remember thinking I need to study e-cigarettes was talking to a veteran who had said, should he try? And I'm like, I don't know. I don't know what they're going to do. I don't feel comfortable telling you to use something that I know nothing about. And then I was like, oh, I can actually study this in the lab and start answering these questions. So it was inspiring. The early generation e-cigarettes were completely not helpful for smoking cessation. So the first, second, and third generation devices, they had very low levels of nicotine, and that's why it's thought that they weren't successful. But the new generation, as I mentioned, they can deliver 59 milligrams per mil nicotine. And so they activate that dopamine reward system like nobody's business. And so they are more successful. So if somebody were to use that to try and quit, they could potentially quit smoking combustibles, but they won't be quit off of tobacco because they would most of them stay on the e-cigarettes. And in worst case scenario, they actually combine the two. So they end up being a dual user. So I always take this data with a grain of salt, but the last two big reviews that came out did say that they were similar to or slightly superior to the nicotine replacement therapies. But then most of them do not report the rates of people who continue on vaping or who become dual users. So keep that in the back of your mind. So when now people come to me and ask about them, that's pretty much what I tell them. I'm like, look, they're about equivalent to nicotine replacement therapies. I recommend using the nicotine replacement therapies. If you are to try using the e-cigarettes, you have to come off them. Do not use both of them together. And when people ask me to quantify harm reduction, so I get pushed on this a lot by reporters, and they're like, well, shouldn't you be supporting e-cigarettes for your patients because they reduce harm? And when you do the math, you're like, well, even if they were to be 80% safer, which it doesn't look like they are, that would still lead to 1.6 million deaths per year. So just, keeping in mind that, it's not, for nothing that you're trading these two vices. Okay, but what diseases will e-cigarette vaping cause? One thing we're worried about is actually some metabolic effects of vaping. This study was also run by high schoolers and community college students. And what this reveals is, so the hypothesis was really fun. So they wanted to know if vaping THC had the same couch potato and munchies effect that smoking regular marijuana had. And so what we found is that those teenagers who were only using THC in the light blue had the same sugary drink, sweet intake, and same level of exercise as the control teenagers in white who were not inhalant users. But we also asked about their nicotine intake, and the teenagers who both used THC and nicotine actually had higher sugary drink and sweets intake. And so that was very interesting to us. And so we're sort of trying to drill down on that to see why that would be, if that's an effect on your flavor receptors. But you would imagine that as a teenager, you drink more sweet drinks and take in more sweets, that you could impact metabolic syndromes down the line. And a survey that we ran a couple years ago that was just published last year, we also looked at sleep, because sleep broadly impacts health. And this was a fun study to run because it actually, people took it all across the world. And we found that conventional tobacco in pink and e-cigarette users in teal really had the same levels of sleep quality as our non-smokers, non-vapors in black. But our dual users of both combustible tobacco and e-cigarettes in purple had a higher sleep latency, which means it took them longer to fall asleep. And on average, that was about 8 to 11 minutes longer for them to fall asleep than anybody else. So they're just laying in bed, hoping to fall asleep for that additional 10 minutes. And so we've been designing some other studies to try and drill down on the impacts of sleep. If you want a more detailed read about all the different things e-cigarettes do, this review that I wrote with David and Jinnah Park at Harvard really covers the lung effects in cellular and molecular detail, and also covers some of the over the body effects as well, as we clearly won't have time to go into all of that. And then the one disease that is specific for e-cigarettes is EVALI. So the e-cigarette or vaping product use-associated lung injury, EVALI. This was a disease entity before 2019. I had seen patients with this, but it didn't have a name at that time. And then in the summer of 2019, hundreds of people were impacted. It led to thousands of people affected with this lung injury. And in the end, it was reported in all 50 states, as well as Puerto Rico, and then other countries started reporting it as well. And then the CDC stopped tracking it when COVID hit, but we still see it at UCSD, and I'll tell you a little bit about how it's changed over time. So the 2019 epidemic was a disease of the very young. So median age of 22, and most of them were under 35. And 82% were vapors of THC. And so there's only a small subset of these EVALI patients, 16, 18%, excuse me, that were vapors of nicotine only. And that's really important because now, in the years since that epidemic of 2019, yes, the cases have dramatically fallen, but we realized that 16 to 18% of cases has puttered on. And that now we're seeing half of those being vapors of THC and half of them are vapors of nicotine. And the reason why I'm harping on this is because the epidemic of 2019 was caused by the addition of vitamin E acetate to THC liquids in particular. So this massive molecule that you can see behind these pictures, it's fine to rub it on your skin, it's fine to take it in through the GI tract, but if you vape it and inhale it, it just shreds your lungs. So it causes acute lung injury. But this chemical is not added usually to nicotine e-liquids. So what's causing the E-Valley cases and the nicotine-only vapors? And so that's where work still needs to happen. And we don't actually even have a registry yet for E-Valley. So it's really from talking to the other clinicians who care for these cases that we're trying to put the pieces together. This was an article that I wrote for JCI, and it was like my favorite article ever because they reached out and they were like, hey, what do you think is going on with E-Valley? Can you write something and we'll publish it? And I'm like, really? Like no data? I just write what I think is going on. And they're like, yeah. And it was so much fun. So I just like wrote this thing and they published it. So it was really fun. So on the left-hand side, this is something that we've actually proven now through e-cigarette studies in mice and in humans, is that the state of the lung is fundamentally altered by the exposure to these e-cigarette chemicals. And so when you look at any of the epithelial cells or that beautiful green alveolar macrophage, the phenotype is shifted. So the gene expression is different, the protein expression is different, and their functionality is different. But if you looked at it histologically, so the H&E stains or radiologically, it looks totally normal. So we're worried about that second hit phenomenon. So when somebody then inhales air pollution or gets a viral infection or changes the flavor of the e-cigarette that they're using so that they're inhaling a new chemical, that can trigger acute lung injury and a pathologic inflammatory response. So that's something that we really do think is happening. In terms of E-Valley, when I'm reviewing cases now, I try and put them into different categories, meaning mild in green, moderate in yellow, red and severe. Most of our data is actually in the red severe cases because those are the ones that are typically recognized and reported out because they're getting like a really deep history. And so we don't know as much about the mild cases, which never come into the hospital, never needed oxygen. And so we don't know what the rates of those are. We have tried to identify biomarkers of E-Valley so that we can have something more specific to guide people when they're trying to diagnose it. And unfortunately, that has not panned out. On the left-hand side, we analyze levels of ESR, CRP, and total white count. And we tried to put it in the mild, moderate, severe cases across the horizontal, but you'll see we had no data for the mild cases. And then for the moderate and severe, the best one was ESR being sky high. The CRP was a little bit mixed and the white blood cell count was mixed. So when people call me and they ask what lab to send off, I'm like, ESR. So it's not very rewarding to still not have a biomarker. On the right-hand side, those are our BAL studies. So we were hoping that there might be a specific pattern of immune and inflammatory cells in the airways of our E-Valley patients. Again, the mild cases don't get bronchoscopies. But you can look at our moderates and our severes, the total absolute neutrophil cell count, we only had one in the moderate case. So in the severe cases, it tends to be higher. The percent neutrophils tends to be higher. The percent macrophages tends to be higher sometimes. So again, we don't have a good pattern to go by. I expect neutrophils to be there based on what we've seen, and that's about it. Okay, so beyond E-Valley, which causes lung injury, what we've really been focused on in the lab for the last year is whether vaping causes cancer. And so really excited to show you guys our latest data. Oddly enough, the first publication from the lab was done in collaboration with Wagon Keko. And in that study, we found double-stranded DNA breaks in cells exposed to e-cigarettes. So our first paper was showing DNA damage. And since I give the DNA damage lecture to the med students, I know that the double-stranded DNA breaks are the worst ones and that they drive more mutagenesis than other ones do. And so we collaborated with Pradita Ghosh to look in the colon. So I mentioned that we harvest the colons of all of our mice. And what we found is that our air mice on the left-hand side have beautiful colonic epithelium, but our mice exposed for 3 months daily to e-cigarettes without nicotine in the middle had massive amounts of inflammation. And then on the right-hand side, our e-cigarette with nicotine exposed mice had inflammation as well. And so when we're brainstorming what is going on here, 2 different pathways come to mind. One where the e-cigarette chemicals, because they go into the oropharynx, that they're actually swallowed and it actually directly impact the epithelium through contact and cause damage. And the second one being that they rapidly enter the bloodstream, about 90% of the chemicals enter the bloodstream once you inhale them, and that they could reach the colon in that way from the interior. So still not sure what that answer is. But when we then started doing studies with APC mice who spontaneously develop colonic polyps over the first couple months of life, we did this work in collaboration with Shumita Das and Ibrahim Saeed. Our air control mice on the top, that's the normal amount of polyps that they develop. And if they were exposed to e-cigarettes for a month, they had many more polyps, and they actually were very similar in number and size to the cigarette smoke exposed mice in the third panel. But the mice that got half e-cigarettes and half cigarette smoke each day had the worst response with the most polyps. And not only did they have increased polyp number and size, but they had increased this epithelial inflammation. So same pattern here where the air mice on the left look great, and then the e-cigarette nicotine mice, the cigarette smoke mice, CS, and then the dual exposed mice, and then their histology score on the right showing just increased inflammation over time. And when we looked a little bit deeper at the proteins, we found increased markers of proliferation as well. And when we looked at DNA repair enzymes and proteins, a couple of them were altered. So decreased base excision repair protein NEAL2 on the top, no change in the base excision repair protein OG1, and then HG2AX is a sign of double-stranded DNA breaks. So this was nice to see evidence of that in the tissue itself instead of in a dish. And then on the right-hand side, when we then took another look at our lungs transcriptomics, we see a lot of the downregulated pathways include DNA repair pathways. So we're seeing these mirror images in the colon and the lung. So the lung that's directly exposed for sure to these e-cigarette chemicals, and then the colon either exposed directly through swallowing or through the bloodstream, both having these effects. So we actually exposed these KRAS mutation mice in collaboration with Mark Onitis and Mark Fuster. We exposed them actually before COVID hit. And luckily, as UCSD was shutting down, we were able to madly harvest these mice and send them to Guanfeng Wang, who's one of the postdocs in Mark's lab. And we actually just finally got the data now after a long hiatus of them just sitting there. And what we found was in the air mice on the left, you see beautiful tiny little adenomas spontaneously arising because of the KRAS mutation. And then the e-cigarette exposed mice with nicotine in the middle, having more of those and larger adenomas. And on the far right-hand side, the e-cigarette without nicotine, the vehicle mice, also having more of the adenomas and larger ones. And then that quantification is just on the far right side. So I hope what I've convinced you of is that we're seeing tumorigenesis in both the colon and the lung, and that it's not driven by nicotine, that it looks like it's driven by the other chemical components in e-cigarettes. And it looks like it's to equal degrees as the cigarette smoke would. And then I've talked about the altered inflammation in the colon. We also use human colon organoids and challenge them with LPS. And in black, the ones that were exposed multiple times to e-cigarettes had a higher IL-6 response. So they had an increased inflammatory response. And then on the far right-hand side, we also infected them with bacteria, and they were less able to handle that challenge. So we see this dynamic of an increased inflammatory response, but a decreased host immune response. So it's a terrible combination. So how do I put this together for the cancer? So on the upper left-hand corner, as I mentioned, we found DNA damage. Multiple other groups now have found DNA damage caused by the e-cigarette aerosols. On the lower left-hand side, showed you lots of data about the inflammatory changes across the body. And then on the red circle on the right-hand side, epithelial barrier breakdown, we've seen that in human airway cells. They have decreased zona occludens and these other tight junctions, and so there's leakiness. And that we've seen that also in the colon as well. And then the new area that we're moving into with the help of Greg Botta and Mark Fuster is trying to define whether the immunomodulation of tumor-associated macrophages, dendritic cells, and then NK cells might also be playing a role. I'm an innate immune person, and so it's been really fun to study neutrophils and macrophages and really seeing how they're functionally altered by the e-cigarettes. But it really raises concern that those macrophages that are around tumors are probably not responding the way that they should. But hopefully in another year, we'll have a good update on that. Okay, so who's convinced that they might cause cancer? All right, we see some hands raised. Hey, goal achieved. And on the human side, we're continuing to get samples from human subjects. So we've done 2 different human cohorts. So in 2016, 2017, we recruited 22 e-cigarette vapors and 12 controls. And we found in the plasma that there were broad increases in inflammatory proteins. And the monocytes in the circulation of those e-cigarette vapors had functional changes. So in this top panel, the black monocytes are from our non-vaping, non-smoking controls. And if you challenge them with LPS, they have this beautiful increase in IL-8 production in the hollow circles. But our blue monocytes from our e-cigarette vapors did not have a statistically significant increase. So they really were kind of quiescent, like immunosuppressed, like not able to respond to this like very powerful inflammatory challenge. And then on the bottom panel, we asked all of our vapors to quit for 2 weeks. Didn't work at all. None of them could quit. They all said that they could. They were like, no problem. And then they came back 2 weeks later and they're like, couldn't do it. Like they had no idea that they were so addicted. But they were all able to decrease their vaping, and they actually had return of function with the response to the LPS with both IL-6, IL-8, and TNF-alpha. So sort of proof of concept that these monocytes are definitely in an immunosuppressed state in the circulation of vapors. We just completed, thanks primarily to Nikita in the lower left-hand corner there, recruiting 100 human subjects for our second cohort. So we have about 75 e-cigarette vapors and 25 controls. And this time mainly because we couldn't recruit just nicotine vapors because it's like impossible. So we have THC vapors as well. We have dual nicotine and THC vapors. And then I want to tell you a little bit about our asthma question, because there's some really interesting signals coming down that indicate that e-cigarettes might impact asthma in a specific way. That asthma work is actually done in collaboration with Vijay, who's at La Jolla Institute of Allergy and Immunology, as well as our own Praveen Akuthoda. From these new 100 subjects, we were able to do transcriptomics on 4 of the sample types. So nasal brushing in the left upper dot plot, and then peripheral blood mononuclear cells in the upper right, sputum lower left, and PAX gene RNA, which is really a good way to broadly assess neutrophil gene expression on the lower right. And what should pop out to you is that the samples from the nasal brushing had the most dramatic changes compared to controls. So that tons of red and the airway epithelium from the nose having gene expression changes in the e-cigarette vapors relative to controls. You see a smattering of changes in the sputum and the PAX gene and the PBMCs, but the big signal seems to be coming from that upper airway epithelial space. And then for the asthma data so far, this is just a teaser because Nick Hogan, one of our fellows, is working on this project with Praveen, where we're trying to drill down on the effects of the e-cigarettes on eosinophils. So we're pretty solid on the effects on monocytes, macrophages, neutrophils, but this is eosinophil specific work. And on the upper right-hand side, this is a pilot data from our new e-cigarette subjects. And the takeaway there is that controls have very low levels of airway eosinophils. These are from the induced sputum. But our e-cigarette nicotine-only vapors in the light blue, and then our e-cigarette nicotine and THC vapors in the darker blue, both had elevations in airway eosinophils. And then on the far right are e-cigarette asthmatics, which not surprisingly have elevated eosinophils. So we're hoping through the work that Nick is doing that we can identify the exact gene expression pathways being hit and figure out like which of the chemicals are driving that. Another area, I mentioned the sex effects in the lung transcriptome. We're also looking in the liver and the kidney, which are susceptible to fibrosis actually. And Howard and Jared were leading this work. So hopefully more to come there. And what I wanted to end on is the data that we have from the brains of mice. And so this, I was sort of stuck for a while with this because I am not a neurologist. I don't typically study the brain, but Arthur Brody, who actually does do that, is interested in studying this in humans. So we're working on a collaboration there. And what these data show is that in the nucleus accumbens, which is in red in that diagram in the upper right-hand corner, when we look just in that spot of the brain and we looked for inflammatory signals and we looked by gene expression and protein, we looked after 1 month, which is a subacute exposure, versus 3 months, which is about like 10 years in human years. TNF alpha, IL-1 beta, and IL-6 were all elevated. And it didn't matter what the flavor was or the duration. We also found elevations in RAGE and HMGB1. And the takeaway there is that the nucleus accumbens is really critical for the dopamine reward pathway, but it's also involved in depression and anxiety and potentially even OCD. And so we're really worried about our young people picking up e-cigarettes, having these neurologic changes, and then becoming more susceptible to depression, anxiety, and other substance use disorders. So you can imagine in mice, like I'm not really set up to answer these questions, but there's some great psychologists and psychiatrists who are working in this field and really hoping to understand a bit more about the impact on mental health. Since I was up here, just wanted to give a shout out to this program. This is a program that TRDRP funded, so the Tobacco Research Disease-Related Program of California. That program is funded by the tobacco taxes. So the money that our tobacco users pay to the state, some of it is sent back to research in dollars. So this program, and you can use the QR code for more information, is great because it has allowed us to recruit scholars from communities disproportionately affected by tobacco. And we put them through a 1-year training program to train them up on tobacco advocacy as well as tobacco research. So we just had our first 3 scholars graduate, Annie, Christina, and Linnell. And our new set of 4, Bianca, Janet, Patricia, and Paulina, are just starting now. So this has been a really fun program to be involved with. And actually, Annie, pictured on the left, she's designed a study that just got IRB approval to try and drill down on that question I mentioned of why do young people switch from e-cigarettes to combustible tobacco, or why do they add it in? Because nobody seems to know the answer. So hopefully she'll know that answer soon. So back to these 5 takeaways. Hopefully I've been successful and convinced you that e-cigarettes hurt the lungs, that they are going to cause cancer. They hit everything across the body. The dual use data coming down the line from other labs is just as strong as what we're seeing in our lab, that 2 different inhalants seem to have a much greater effect on the body. And then if your patient is inhaling something, you need to really ask them about every possible thing they could be inhaling, because the % rates of dual and triple use are very high. I do quite a bit of work in policy at the national level with CHEST and sometimes through ATS. And these are the three big areas that we work in nationally and locally. If we could limit the amount of nicotine in the e-cigarettes, we think that that would make a big impact. In the UK, they have not had the epidemic of e-cigarette use in young people that we've seen. And one of the reasons why, because they have exactly the same e-cigarettes, except they're limiting their nicotine to 20 milligrams per mil, whereas the limit in the United States is 60 milligrams per mil. And we think that that made a huge difference in that they just aren't giving them the high, they're just not as appealing. So that's one opportunity. A second one is that if we could ban flavors that target children, women, and people of color, that would be a huge win, and that we would be protecting these populations. And then, of course, banning sales near schools. So I definitely have people text me pictures of tobacco and vape shops that are like right next door to middle schools, and it's just depressing. And finally, messaging that we should share with our friends and family and colleagues and patients. The PGVG, so just the base components of e-cigarettes, are not safe to inhale, not even talking about the nicotine effect. Nicotine itself, super addictive, and that use of one has a gateway effect to using another. Thank you to the tons of great collaborators, and then the lab crew. I couldn't quite wedge everybody in, but they're amazing to work with. It's the best part of my day to meet with them and talk about these data. And thank you to the NIH and the VA and the TRDRP for the funding. And we do get out and do some fun stuff. So there's some dinner parties, and this one in the upper middle is us at the YMCA last week doing an outreach session with teenagers. And at the very bottom is like, it's also really great to have so many amazing women in pulmonary critical care across the county. And so we enjoy our time with San Diego lung ladies. And thank you. I'll take any questions. Okay. Oh, Judd?
Really good, Laura. I was just thinking, when you mentioned the different flavors of peppermint, menthol, and THC, these are all airway receptor irritants. And I wonder if you think about the irritant receptor system and how that might impact the inflammatory system because of these different signals you're getting.
I think that's a great idea. And for years, I didn't enter the flavorant space. I think partially because I was trying to keep the model really simple. Because as you add those in, then it's all these pathways that I've never studied before, but I definitely think that it is a complex set of signals. So I do think that that is probably playing a role, and that might be really helpful to see how they interact. So great question.
Jason, you and Vlad have a lot of
Okay, I'll talk to Jason then.
Thanks, Laura. That was really interesting. A lot of information. Anybody in the audience here? You get preference. We have some online questions. Positive reinforcement. If you show up, you get your questions answered.
Thank you, Laura. Fascinating talk. You showed early on a dramatic sex difference. Now, have you, in your additional studies, did you separate males and females? And is there a difference clinically too?
I think there is going to be, and I think that that's going to be one of the reasons why epidemiologically we might not see consistent signals, because we're going to have to look by sex, and then we have to look by flavor. And so the number of people is going to be really small that fits into those buckets. I am really excited about the new set of human subjects, because of the 75 vapors, over half are women and the other half are men. And so we should be able to look at sex effects across these different samples. So that's definitely a major thing that we're going to look for. And of course, we did a deep phenotyping of each of these. So we'll also have that flavorant concept. But because there's so many different flavors, we're usually not powered to be able to tease that out. But I'm hopeful that we can see some more of the sex differences.
Go ahead.
Great talk as always. This is mostly a conjecture question for you, but do you think that there is a risk for secondhand vehicle exposure in the same way that there's secondhand smoke exposure for tobacco products? Or do you think that because the vehicle is so dense and heavy that perhaps it may not be as big of an issue? And is there any research in that area?
Yeah, no, great question. I think my favorite study in that area was they put a vapor in a room with a non-vapor, and then they measure the levels of the chemicals in the bloodstream. And there was like a decent secondhand exposure. So it was about 25% of what the whole, the primary user was seeing. So, the e-cigarette aerosols are primarily droplet, and so they do sink much more than cigarette smoke, which goes up and out. So we are more worried about things that are lower down. So children and animals are having higher exposures than probably other adults hanging out. We also particularly worry about babies and pets because these aerosols are so heavy, they sink down and they create an oil slick on the surfaces. So if anybody wants to witness this, you can come to the lab and see what the stuff looks like after e-cigarette exposure, and you just put your finger on it and it's gross. It's just literally like an oil slick. And because the propylene glycol is so good, it actually you have a pretty high transdermal absorption. So it'll be a different type of exposure. So it'll be less inhalant and more dermatologic. But they have seen that the nicotine gets transdermally taken up, and so that pets and children are getting it transdermally and through the inhalant pathway. Great question.
Thank you, Dr. Crotty-Alexander. Was super interesting. I was wondering, I think one of the mechanisms you mentioned earlier is kind of injury caused like through kind of, spread by the blood circulation system. And then like kind of interesting with the gut, changes in the epithelium. I was wondering if we have already seen like kind of the effects of e-cigarettes on the cardiovascular system, like the way we see it in like combustible cigarettes.
Yeah, again, a great area for research. And we have looked in mice at blood pressure changes, heart rate variability changes. And we hadn't really seen consistent findings outside of subtle increases in the diastolic blood pressure and some slight tachycardia. That looks like it's repeated on the human side. So when they do test those in human vapors, there is that sort of acute tachycardia and response to the vaping that is similar to combustible tobacco. So the thought is that they are going to have cardiovascular effects. I would say that the most powerful data has been in the systemic endothelium. So when the endothelial researchers have looked at the peripheral vessels, they've seen very dramatic changes in the spasm and in the levels of inflammation and oxidative stress that suggest that there is a really powerful endothelial signal going on. So, and I do think that that's actually some of the most powerful data, most compelling data that is out there. So yeah, great question.
Okay, we'll take some questions from the chat from Margaret Mohammed. Thank you, Dr. Crotty-Alexander. The science is convincing, but how do we translate that to the general public? And how do we affect public policy to reflect the understanding that e-cigarettes are harmful to health?
Yeah, absolutely. Great question. And actually the TRDRP program is part of the grant when you submit it is how are you going to engage with the community? How are you going to get your data to the public? And luckily, I've been doing a lot of it already. So usually if any group asks me to speak, I say yes. So particularly if it's schools or, as I said before, the YMCA, just to try and get the word out there and just share the data that we know. So that's been one way, is just to directly interact with groups that are asking for that information. Other ways are really, posting on Twitter and Facebook. And if reporters reach out to talk to me, I always say yes as well. And I'd say 90% of the time it's really educating them on the field, and they use like 1% of what I say, but I'm hoping that by educating them more broadly that they'll be thinking of that as they write other stories. I think it's very hard to convince the public when you have primarily mouse data and cellular data. So I try and really simplify it and to like very focused key points.
I think an important point on that in terms of the human effects also is that they haven't been around for that long, right? So we don't even know what we're actually going to see in another 10 or 20 years, right?
Yes.
Based on the mouse data. Okay, we have another question regarding the effects on sleep latency, which was increased. They want to know how was it measured? And did they recruit human subjects to a specific location and then do a sleep study, or was it mouse and extrapolated to human?
No, great question. We actually wrote a grant with Atul Malhotra to do like in-house sleep lab studies, because I think we do need to use human vapors and very controlled settings to define these sleep effects more. The data I shared with you was all self-reported, sort of based on the validated sleep questionnaires that are out there, but it is self-reported data. So it's definitely like the weakest data that there could be. But we did run it 3 different times and got very similar findings, particularly for the dual users, and that the women in particular were more affected than men. But yeah, we need to do the real in-house sleep lab studies for that.
Okay, you've generated a lot of interest here. Lynch syndrome requires an acquired second allelic mutation to generate DNA mutations and ultimately cancer. As your data demonstrates, mutations in AKT mutated mice leads to enhanced tumors. Are there similar drivers in humans that may be necessary for oncogenesis? You probably don't know that yet.
I don't know that yet, but just mentioning the Lynch syndrome. So we just, I finished Foundations of Medicine. I co-direct that with Erica Lynn. So it's the first 5 weeks of medical school. And we totally had on the final like Lynch syndrome questions. I'm having flashbacks to that. Yeah, I think that there are numerous pathways that we really need to go deep on. And so what we're proposing is to, be more specific in these pathways. And what we want to do is take like 10 adenomas from each of those mice and do whole DNA sequencing, exome sequencing, to define the exact mutations happening in each of the different exposures to see if there's more of an impact on specific pathways driving tumorigenesis.
Okay, from Dr. Moore, how can we stop initiation of inhalation of such substances? And then just a general question of how do you do it all?
Thanks, Allison. If I knew the answer to your first one, then we'd be all set. Yeah, I think it's really hard. I think I really focus on teenagers and young adults. And so I really try and just share the data and don't make judgments, and I don't tell them what to do because that usually works better for teenagers. Just, try and have them understand what they're doing so that they can make informed decisions. But all of us, choose to do things that are not great for us. And so, I feel like you just have to try and help people understand all the downstream effects that might happen and then allow them to be a free-willed person. And how do I do it all? Well, I don't really know anymore.
We have a lot of good people, actually.
Yes, thank you. That's
From
how we do it.
From Dr. Lewinsky, are the sex effects hormone mediated? For instance, what is the phenotype in trans feminine subjects on hormone therapy?
That's exactly where we want to go. Again, like I've seen these sort of sex effects, but that is not an area of research that I am an expert in. And actually, Alexia Perryman in our lab has been looking more into that and looking. So there's a great mouse model that we could use that has XX, XY for both female and male. And we want to do hormone therapy as well, because we have no idea why it's happening. And we definitely need to drill down on that. So if you have suggestions, feel free to email.
Okay, from your division chief who's watching this, great talk. Are we able to get advanced info on new vaping equipment and substances? Always feel like we're playing catch up.
Yeah, I'm hopeful, which is a weird thing to say, but the e-cigarettes and the liquids evolved so dramatically from 2007 to 2016. So, within those 9 years, we went through 3 generations. So we've been at the 4th generation since 2016. And I'm kind of hopeful that the 4th generation is so successful at what it does that it will stay about the same moving forward. And that like the positive side of Juul is that they like maximize nicotine delivery. And so I'm kind of hopeful that again, the e-liquids also have sort of hit a level where they don't need to be evolved as dramatically, because it does make our lives easier. Because in mice, we started by exposing them to 1st gen, and then we moved within 2 years to 2nd gen, and then within 2 years to 3rd gen, and then within 2 years to 4th gen. And so it's been kind of nice to actually stick with the 4th gen for the last 5 years. But in terms of anticipation, it's really that running the surveys locally and nationally helps me understand what the patterns are and seeing them change before they're reported out by the CDC, the national levels, or even locally, so that we can see like where is the shift happening, like which of the devices are becoming more popular, which flavors are becoming more popular. And that has helped me try and make sure that the research that we're doing is most relevant to current e-cigarette users.
Okay, do you have a slide set you can share with middle and upper school teachers, which is not a bad idea?
That's a great question.
Yeah.
I will have one of my awesome lab team members make one, but that's a great idea. And we could actually just post it on our lab website. So thank you for that. That's a great suggestion.
Any other questions? Yeah, otherwise I have one. So the remarkable thing is how normal the histology is when you actually look at the lungs. Do you think that over time that will change again, because it hasn't been around for so long? Certainly the DNA damage and like bleomycin does DNA damage, does fibrosis. You're not seeing that here. What do you think will happen when people have been doing this for a long period of time?
Yeah. And in mice, some people have gone longer, like 4 to 6 months, which is more like, multiple decades. And some of those people have seen emphysematous changes. But I think that I come back to that idea that because we've immunomodulated the entire lung and it's going to respond differently to second challenges, that I think that the changes we see in humans is going to be much more dramatic than in our mice that we're keeping in a very controlled environment and really avoiding all second hits as much as possible. And I know some groups are actively adding in the second hits to see like how much worse it makes the lung disease. And I'm really fascinated to see that data.
Because I think actually starting now to do some longitudinal studies would be really, I guess the registry would do that too, but
Yeah.
longitudinal studies of PFDs and things like that. But
So we are trying to, the 100 that we just recruited, because we started in May of last year, we've already started annual visits, because I feel like the longitudinal data is going to be way more interesting and insightful than the pinpoint view because of all these different factors. And we are doing just basic spirometry, the one-held spirometry, and then response to bronchodilator after their induced sputum. So hoping that that gives us some more data that we would love to do, like the real lung function test over time. And the high-end xenon imaging combined with MRI that does the physiology of the vessels and the airways. So we've seen like signals there, but longitudinally, we think that they would be more powerful and that we detect more.
any remaining questions? That was wonderful. Generated a lot of discussion and interest. So thank you.
Thank you.
Good job. Now you can breathe again.
Yeah, exactly. I wanted to tell you I didn't want to do it because I kept thinking it was inside, but it looked good. I almost took a picture this morning. Yeah, I'll send you a picture because it's like, Tom took over caring for it. Which is the only reason that it's alive.
I'm so impressed. So I'm still alive in your house. That's good.
Yeah,
That's wonderful.
one of them flew and the other was about
You
to.
know that I've coordinating knocked out mice develop tumors.
Oh, I didn't